Hello guys, welcome back to my channel. I'm Joseph, and today we are going to talk a little bit about modeling complex biological system. But before we continue, if you find my videos and my channel helpful, please feel free to leave a like and subscribe, and leave a comment down there to tell me what videos you would like me to make in the future. First and foremost, this is a work that was done by a former graduate student of mine, Furnas, in collaboration with Hyun Seok Song. What I'm going to do in this particular video here is to go through some of the key concepts underlying the modeling framework and why we came out with this framework and what are some of the problems that the framework can help to solve. So let's first just define the problem. So nowadays we talk about using or utilizing the potential of biomass to produce valuable chemicals. And so we need to understand that in biomass itself, one of the major constituent is the cellulose, and cellulose is a polymer. All right, and this particular material of cellulose is very complicated because when we look at at a very fine scale, we notice that cellulose consists of polymers that are linked by glucose monomers, and they have varying degree of polymerization, right? They are distributed quantity. So, but when you zoom out a little bit, then you see that the aggregation of these cellulose polymers would form cellulose fiber, and then fibers can aggregate together, and you would have different regions of the cellulose. For example, it can exhibit a crystalline region, and it can also exhibit an amorphous region. So when you have all those stacking together on a much larger or more macroscopic scale, then we can view cellulose as a particle. For example, if you actually grind the biomass, right, you put it under some milling or grinding operation, then you would see that cellulose is a particle. But within that, to the very low level, or to the very microscopic scale, they exist as polymers. So first and foremost, the ability to actually process this cellulose is a non-trivial problem, right? It's very complicated. So in the event that we really want to use microorganism to actually convert cellulose into valuable product, what the microbe needs to do is to actually what the microbe needs to do is to excrete the necessary enzyme that can actually break down this complicated cellulose material into fermentable sugar or simple sugar such as glucose. And then when you have these simple carbon sources, the microbe can assimilate them and use them or metabolize them to produce valuable product. So this is a closed loop system, right? This is a very complicated closed loop system because when there is no simple carbon sources, the microbe needs to excrete cellulase, this particular enzyme, in order to break down the cellulose. And when the cellulose is being converted into simple sugar, when simple carbon sources are being more abundant, then the microbe will be able to assimilate this for its own metabolism. So there are two key things, two critical points here that makes this process complicated to understand. The first is that the polymeric substrate itself, cellulose, is a very complicated material. It's a distributed quantity, so we need to be able to understand this, right? Because every cellulose is going to be different. Because when the cellulose has different chain length, then you talk about number average molecular weight, you talk about the weight average molecular weight, polydispersity index. So those are things that are a property of polymer systems. But on the other hand, then you have this microbial metabolism. In the microbial cell itself, you have a complicated bunch of metabolic network. So the microbe, in response to this complex polymeric substrate, it will regulate its own metabolism in order to decide, mainly in this context, when to produce more enzymes to break down the cellulose, and when to actually start producing enzymes internally in order to metabolize the simple sugar that is being produced in order to maintain its own growth and other metabolic function. So the steering of the microbial metabolism in response to environmental changes or diversity here is also something that we need to take into consideration. So these two key things here, complex polymeric substrate and complex metabolism of microbial system. So these are the two key things that we need to take into consideration when we try to actually understand consolidated bioprocessing, which is a process that has the enzyme produce its own, has the microbe, where the microbe produce its own enzyme to break down cellulose, and at the same time assimilate and metabolize the simple sugar that is broken down from cellulose for its own metabolism in order to produce valuable biochemicals that we want. So this whole process here is a complicated closed-loop dynamical system, which our aim here is to derive and to establish a framework, a modeling framework that can actually allow us to abstract the principles behind such system. So in order to solve this problem and understand consolidated bioprocessing of cellulose, we created a framework called the UC-PBM, the Unified-Cybernetic Population Balance Model. So basically, in a nutshell or simple words, what we do is that we use population balances in order to model the cellulose as a distributed quantity, which means that we actually took into account the degree of polymerization distribution of the cellulose substrate or cellulose particle. So we actually try to model this particle explicitly, and then we use the cybernetic modeling approach, cybernetic models, which is shown here in green. We use that model to actually model how the microbes excrete the cellulase or the enzymes that can actually break down cellulose polymers into simple sugar, right? So we model how the microbe actually regulate its own metabolism in order to decide to actually excrete this enzyme to break down the simple sugar, and at the same time also have to balance its own growth for its own survival, right? Because when there is no food, we hypothesize that when there is no food around, when you only have cellulose as a particle which the microbe cannot immediately assimilate, then the microbe has to decide to produce more cellulase in order to break the cellulose down to simple sugar. And once the simple carbon sources are abundant, then the microbe has to steer its own metabolism in order to pick up the simple sugar and start to use them for cell growth. So all this phenomena of metabolism and the breakdown of cellulose to cellulase, we solve that by employing a coupling between the cybernetic model and the population balance framework. Let's first take a look at the saccharification component of the UC-PBM framework, where we utilize population balances to model the breakdown of cellulose particle or polymer by the enzymes. So if we assume that the cellulose chain of varying degrees of polymerization in the cellulose particle is being distributed uniformly throughout the entire cellulose particle cross-section, and we use this assumption to build the model, we realize that no matter how we build the model, we are not going to be able to predict the experimental data, which is shown by the symbols here. So even if we manage to forcefully fit this, which is not something that we want to do, we would often realize that we will arrive at a locally valid model, which is not very useful. So in the experimental data of enzymatic hydrolysis, we would often see that there is a rapid phase of hydrolysis at the beginning of hydrolysis, and then followed by a slow phase of hydrolysis. So what we have done differently is that we hypothesize that because of the pre-treatment on cellulose particle, all the cellulose polymers on the surface of the cellulose particle would frequently already be destroyed, right? Such that the shorter cellulose chain and the mainly amorphous region lies on the outside of the cellulose particle, whereas the longer cellulose chain, which is mainly crystalline, will be deposited in the inner side of the cellulose particle. We call the outer layer the penetration zone, and then we call the inner layer the internal zone. So by using this hypothesis, we notice that we are able to predict experimental data with a universal set of kinetic parameters, which means that we do not have to refit the data for different experiments and making them just locally valid. We also tested this with various experimental data in the literature, and we found that the model was able to predict decently the experimental data. So that is in a nutshell what we've done with the population balance part of the UC-PBM. Let us now move on to the cybernetic modeling component of our UC-PBM. So let's just take a look at a simple example whereby you have a simple metabolic pathway where substrate 1 can be used to produce biomass, and substrate 2 can also be used through a simple metabolic pathway to produce biomass. So the main idea is that the cell only has finite resources, and in order to trigger either this pathway number 1 or pathway number 2, the cell needs to allocate resources in order to induce the production of either enzyme 1 or enzyme 2 in order to trigger this pathway, in order to decide which pathway is being triggered. So after the enzymes are being induced or repressed and produced in the cells, we also have another control by the microbe, whereby the microbe is also able to activate or inhibit the existing enzymes in the internal of the cell. So there are two levels of control here. First is the regulation of the enzyme synthesis. Number 2 is the control of enzyme activity. So the cybernetic modeling approach basically says that the microbe is an optimal strategy whereby it is able to carefully control these two levels of control in order to respond to environmental diversity. And all this is being done by fulfilling a certain objective. Let's say, for example, in order to optimize the growth of the microbe, or in order to optimize the survival, the microbes needs to be able to cleverly manipulate its own metabolism in response to environmental diversity. So that's the basic postulate of the cybernetic modeling approach. However, in reality, for a consolidated bioprocessing of cellulose, in the microbe itself, the metabolic pathway is going to be very, very complicated. Right? It's not just a simple pathway like this where you have just two pathways. In reality, the pathway can be very complicated. And so how do we incorporate the information on metabolic pathway into our modeling, cybernetic modeling? Now we use the idea of the elementary flux mode to do this. Let us now consider a more complicated metabolic pathway. So if we look at this slightly more expanded metabolic pathway, traditionally, in order for us to model the metabolic pathway, we need to go and do experiments and, if possible, measure the data that will allow us to estimate the kinetic parameters of each reaction contains here. And that is obviously a very laborious effort. So it's not easy to do this. So we utilize the idea of the elementary flux mode in order to simplify the metabolic modeling. So basically what the elementary flux mode say is that given a metabolic pathway that looks like this, can we find out the number of non-decomposable pathway that exists? For example, this can be decomposed into elementary mode 1, elementary mode 2, and elementary mode 3. So you will find that in elementary mode 1, the substrate that is being uptake will be used to produce product number 1. And in elementary mode 2, the substrate will be uptake to produce product number 2. And elementary mode number 3, the substrate will be uptake to produce the product number 2 too. And this elementary mode here is sort of like the idea of an elementary reaction in our reaction engineering, whereby you cannot simplify anymore this pathway. For example, if you knock out any reaction in between in the elementary mode, that entire elementary mode will be discarded and it will not exist anymore. So can we use the idea of elementary flux mode and to integrate it with the cybernetic modeling approach and use it to model microbial metabolism in a more efficient way and make the model more predictive? And this has been done. This was done by Hyun Seop Song and Ramkrishna in their work on lump hybrid cybernetic model. So in this work where we built the UC-PBM, we are simply just going to be using the idea of LHCM, the lump hybrid cybernetic model, and we couple it with population balances, and we provide the linkage of the enzyme, the cellulase that is being excreted by the cell to break down the cellulose polymers, and we just provide the linkage to complete this closed loop in order to model CBP. In order to test the UC-PBM framework, what we have done is that we took the metabolic network that we can find from the literature. The metabolic network for this particular species called Clostridium thermosalum is a genome scale metabolic network, and we use this network information and enumerated the elementary flux vector in this case because we applied some constraints on the elementary flux mode. And so we enumerated that and used this information to build the UC-PBM framework, which we couple the cybernetic modeling with the population balance approach. So in the process of building the model, we only use the data of every cell versus time, which is shown here as the first panel. This is the utilization of cellulose, the change in the concentration of cellulose over time during fermentation by the particular species that we pick. So we only use this to pick the to fit the data, and the rest of the data were indicated as open symbols because we use this mainly for validation. So bear in mind, we only use the data from every cell transient in order to build the model, and we found that our model was able to predict the rest of the metabolite really well. It was able to predict the biomass growth really well, and it was also able to predict the excretion of the enzymes that the microbes use to break down the polymers into simple carbon sources. Of course, there are a lot of other information that we can glean from this model, such as the regulatory dynamics of the microbes itself, right? Which pathway is being triggered in general. But that is not if that is not enough, we also took another set of data from the literature. Again, we only fit the model with the every cell concentration over time, and nothing else has changed. As far as the kinetic parameters are concerned, the rest are all universal parameters. And in this second case study here, we actually use the model to predict the transient of many other metabolites of interest. For example, here we have, this is for cell growth, and then we have ethanol, we have lactate, we have formate, acetate, CO2 release, and the cellulose concentration. We have all these metabolites here, or the external enzymes that we intend to excrete, and just by fitting the data against cellulose data itself, the model is able to predict all the other metabolites really, really well. And this actually showed us the promising usage of the UC-PBM framework to study consolidated bioprocessing of cellulose. There are many other things that we can pull out from this UC-PBM framework in order to help us understand microbial physiology better. But to summarize, the UC-PBM takes two critical features of importance, that is inside and outside of the cell, in order to model consolidated bioprocessing of cellulose. That is all for this video, and thank you for your time. Please feel free to check out our paper on the UC-PBM in the description below, and I'll see you in the next video.